Hello my dear students, today we are going to explore the ultrastructure and composition of the plasma membrane, one of the most fascinating topics in cell biology. This delicate boundary is not just a barrier, it's a dynamic structure that controls what enters and exits the cell, while also helping in communication and recognition. Let's dive right in. Basic structure of plasma membrane or cell membrane or plasma lemma. The plasma membrane or cell membrane is mainly composed of lipids, proteins and carbohydrates. Carbohydrates are basically present as glycoprotein and glycolipids. Its fundamental structure is a lipid bilayer, which is the two layers of lipids arranged tail to tail. Lipid molecules are amphipathic molecules, so they have a hydrophobic tail which repel water and an hydrophilic heads which attract water molecules. This arrangement forms a selective barrier to polar molecules and ions. Types of membrane lipids. Membrane lipids are of three types: phospholipids, glycolipids and sterols. Phospholipids are made up of alcohol, which can be either glycerol or sphingosine, fatty acids, phosphate group and another alcohol. Two major types include glycerophospholipids or phosphoglycerides. This is the image showing the basic structure of the glycerophospholipids or phosphoglycerides. This is the glycerol moiety, CH2OH, CHOH and CH2OH, and two fatty acid is esterified with the glycerol moiety. Here the above one is the saturated fatty acid chain and the second one is the unsaturated fatty acid chain. So there is a kink, there is a presence of double bond, and the third carbon of the glycerol is attached with a phosphate group, and there is an head group substituent named as X which is attached with the phosphate group. This X group can be varied. This is the name of X and the formula of X, and this is the name of glycerophospholipid. Glycerophospholipids or phosphoglycerides are the most abundant lipid molecules found in plasma membranes. Among them, phosphatidyl choline, commonly referred to as lecithin, is the most prevalent glycerophospholipid. Choline is neutral phospholipid. This is phosphatidyl. This is the structure of X, and this is the name of X choline, and the name of glycerophospholipid is phosphatidyl choline. If the structure of X is H, then the name of the glycerophospholipid will be phosphatidic acid. If the name and structure of the X is CH2CH2NH3 or ethanolamine, the name of the glycerophospholipid will be phosphatidyl ethanolamine. If the name of the X group is serine, then the structure will be phosphatidyl serine. If the name of the X is glycerol, then it will be phosphatidyl glycerol. If the name of the X is phosphatidyl glycerol, then the lipid will be cardiolipin. If the name of the X is myoinositol 4,5 bisphosphate, this is the structure, then the lipid will be phosphatidyl inositol 4,5 bisphosphate. Now we'll look about sphingospholipids. Sphingospholipids are a class of phospholipid that contains sphingosine, an amine alcohol, instead of glycerol. They are composed of sphingosine, a fatty acid, a phosphate group, and alcohol attached to the phosphate. Sphingomyelin is the most abundant sphingophospholipid. At neutral pH, the polar head groups of phospholipids may vary in charge. No net charge, phosphatidyl choline and phosphatidyl ethanolamine. Net negative charge, phosphatidyl serine and phosphatidyl inositol. Net positive charge, which is very rare in phospholipids. This is the another structure of the sphingosine moiety, and the sphingosine moiety, there is second carbon of the sphingosine moiety which is bonded with the fatty acid chain through esterification reaction through amide bond. This is the amide bond, and with the first carbon of the sphingosine moiety, and X is attached, and head group substituent X is attached. The structure of X can be varied. If the X is H, then the name of the sphingolipid will be ceramide. If the X is phosphocholine, name the sphingolipid will be sphingomyelin. If the X is glucose, name of the sphingolipid will be neutral glycolipid or glucosyl cerebroside. If the name of the X is di, tri or tetrasaccharide, the name of the sphingolipid will be lactosyl ceramide. If the name of X is complex oligosaccharide, then name of the sphingolipid will be ganglioside GM2. Glycolipids. Glycolipids are the fundamental lipids with carbohydrate, which can be either monosaccharides or oligosaccharides, covalently attached to lipid molecules. They can be derived from glucose or galactose. Cerebrosides is a special kind of glycolipid, which is the simplest glycolipid, and it contains a single sugar residue, which can be either glucose or galactose. Gangliosides are more complex glycolipid. They contain branched chain with up to 7 sugar residues. Glycolipids are located exclusively on the outer leaflet of the plasma membrane. Sterol. Sterols are structural lipids found in the membranes of most eukaryotic cells. The defining feature is the steroid nucleus, made up of 4 fused rings. The 6 carbon rings are one 5 carbon ring. The steroid nucleus is nearly polar and highly rigid because fused rings prevent rotation around CC bonds. Cholesterol. Cholesterol is a type of sterol. It is the primary sterol in animal tissue. They are also amphipathic in nature. It has a polar hydroxyl group at C3. It has a non-polar hydrocarbon region, which is the steroid nucleus plus hydrocarbon side chain at C17. Its overall length, that is of 16 carbon fatty acids, is fully extended. Other sterols include stigmasterol, which is found in plants, and ergosterol, which can be found in fungi. Bacteria cannot synthesize sterols. Some species can incorporate external sterols into membranes. Biosynthesis of sterol. All sterols in eukaryotes are synthesized from 5 carbon isoprene subunits, and these subunits also build fat soluble vitamins, quinones and dolichols. Role of lipids in membrane. Membrane fluidity. Lipids maintain the fluidity of biological membranes. No covalent bonds between lipids allow free movement. Flip-flop movement between monolayers are very rare. Lateral diffusion within the same monolayer, the very rapid lateral diffusion can occur, which is approximately 10 to the power 7 times per second. Lipids can also rotate around their long axis. Hydrocarbon chains are highly flexible, especially near the center of the bilayer. This is the image, first image showing the lateral diffusion. Here the lipid molecule move in an artificial lipid bilayer, approximates that occurs in cell membrane. Flip-flop, which is rarely occurs. Transverse flip-flop, this is the rotational movement or lateral movement. Transverse diffusion, flip-flop, which is very slow. The lipid molecule can move, can change their lipid bilayer, and lateral diffusion, they can move side by side, which is very rapid. This is fluidity. Unsaturated hydrocarbons, when present with their kings, they maintain the fluidity of the membrane. But if the unsaturated hydrocarbons is packed close together, the saturated hydrocarbon, they make the membrane viscous. So effect of what is the effect of unsaturated hydrocarbons? Double bonds in unsaturated fatty acid chains prevent tight packing. So these increases the presence of double bond or presence of unsaturation within hydrocarbon chain or fatty acid, increases the fluidity of the membrane or phospholipid bilayer. Which is the role of sterol? This is the image showing the role of sterol in the fluidity of cell membrane, double layer of phospholipid bilayer, and the red colored is the cholesterol. It disrupts the close packing of the phospholipids and helps regulate membrane fluidity and important for cell membrane fluidity. Basically, it provides flexibility and mechanical stability. It aligns with their hydroxyl groups near phospholipid head groups. Rigid steroid rings partially immobilize the hydrocarbon chains near polar heads but keep rest flexible, prevent phase transition by inhibiting close packing of hydrocarbon chains. Specialized lipid, they are inositol phospholipids which are crucial for cell signaling, glycolipids which are very much vital for cell recognition, synthesis and transport. Lipids are synthesized in the endoplasmic reticulum. They first incorporated into cytoplasmic surface of membrane. They are translocated to outer monolayer by lipase enzyme. Asymmetry of lipid bilayer, outer surface rich in carbohydrate group, glycoprotein and glycolipid, inner cytoplasmic surface negatively charged due to unsaturated fatty acid. They result in an asymmetric organization. This is the asymmetric lipid bilayer. What is the dynamic nature of membrane? Plasma membrane can form vesicles by budding. Vesicles can fuse back into the membrane, which is a reverse process. Now we should study membrane proteins, types and organization. Based on the interaction with the lipid bilayer in the membrane, proteins can be of three types. Integral membrane protein, which is tightly associated with the membrane lipid bilayer. This is the integral membrane protein. It can be removed using agents that disrupt hydrophobic interactions like detergents, organic solvents and denaturants. This is the peripheral. This is the second type. This is the peripheral membrane protein which can interact with the lipid bilayer via electrostatic forces and hydrogen bond. It binds to the hydrophilic domain of the integral protein or polar head groups of lipid. We can find in the image the peripheral membrane protein attach the head group of the lipid. It can be detached using mild treatment like high pH carbonate solution. It functions, it regulates membrane bound enzymes, restrict mobility of integral proteins by anchoring them to intracellular structures. Third one is the lipid anchored protein, which is covalently linked to lipids such as long chain fatty acids, isoprenoids, sterols and GPI, glycosylated derivatives of phosphatidylinositol. These lipids act as hydrophobic anchor into the bilayer. A single hydrocarbon chain provides weak anchoring stability often achieved by multiple lipid attachment. This image also showing the position of integral protein or transmembrane protein, and this is the peripheral protein, and this is lipid anchored protein. Lipid anchored protein is also a type of peripheral protein, but the peripheral protein is attached with the lipid bilayer through electrostatic forces and hydrogen forces, which are the non-covalent bonds, but the lipid anchored proteins bind with the lipid bilayer of the membrane with the covalent bond. Now we should study the structural organization in membrane. On the basis of structure, the membrane protein can be of two types: single membrane spanning protein, which are again of two types, group 1 protein and group 2 protein. When N terminal is outside the cell and C terminal is faced towards the cytoplasm, the protein will be called as group 1 protein. This is the single membrane spanning protein. This is the single membrane spanning protein, but it can be group 1 and group 2. In case of group 1 protein, the N terminal face outside and C terminal face the cytoplasmic side. And in case of group 2 protein, C terminal moves outside the cell and the N terminal moves the cytoplasmic cell. But group 2 protein is less common. And now we study the multiple membrane spanning protein. This is the multiple spanning protein, multiple membrane spanning protein or multi-spanning protein. It can be odd numbered hydrophobic region. These are the three hydrophobic region. So N terminal and C terminal on opposite sides, but it can be even numbered hydrophobic region. Then N terminal and C terminals on the same side. The image showing the number of these multiple membrane protein contain the 4 hydrophobic bond. Role of membrane proteins: structural proteins. They are highly lipophilic from the primary backbone. It form the primary backbone of the plasma membrane. They provide mechanical strength. Integral proteins, they are capable of lateral and rotational movement in the lipid bilayer of the cell membrane. They cannot undergo flip-flop or transverse movement. Transport proteins like permeases or carriers, they facilitate movement of substances across the membrane. Transport proteins are of two types. Carrier proteins, they act as mobile carrier. They selectively bind and transport molecules such as glucose. And channel protein, they form open pores in the membrane. They allow free passage of molecules of suitable size. This is the channel protein and this is carrier protein. Membrane carbohydrates, structure and types. They exist as short chain of oligosaccharides which can be unbranched or branched. They are located mainly on the external side of the plasma membrane. They covalently linked to lipids. It can be glycolipids, and proteins, it can be glycoproteins. Glycoproteins are the sugar residues are attached in two ways to the hydroxyl group of serine or threonine. They are O-linked oligosaccharides, and to the amide group of asparagine they will be N-linked oligosaccharides. Common sugars associated monosaccharides, it can be D-glucose, D-galactose, D-mannose, D-xylose, D-fucose and L-arabinose. L-fucose and L-arabinose. And sugar derivatives are N-acetyl D-glucosamine, N-acetyl D-galactosamine, N-acetyl muramic acid. Glycolipids, carbohydrates attached to the glycerol membrane of lipids via glycosidic bonds. If this video helped you understand the ultrastructure and composition of plasma membrane, don't forget to like, share and subscribe to stay updated with membrane biology lessons. See you in the next class. Thank you.